Sorry, the recording got cut off prematurely. So let's continue with enzymes, part two. Sometimes enzymes require an additional molecule or mineral attached to it. So the enzyme itself, in this case, is called the apoenzyme. Oops, sorry about that. It's called the apoenzyme. So that's just the protein. And it becomes more active if you bind a coenzyme or cofactor to it. The difference between a cofactor and coenzyme is a cofactor is inorganic. Something like magnesium might be a cofactor, and then a coenzyme is something that's organic, and most of these are going to be your vitamins, vitamin Bs and stuff like that. So they're organic. Either way, the apoenzyme becomes a holoenzyme when the cofactor or coenzyme is bound, right? So it's fully functional now. It's a holoenzyme that can bind its substrate. For enzymes that require control, right? We want to make sure that our enzymes are functional when we want. The cell can also slow down enzymes or stop enzymes from working with other molecules they make. So to slow down enzymes, this is called enzyme inhibition. It basically just blocks them from working in some way. So there's two ways to slow down or inhibit enzymes from working, and that's competitive inhibition and non-competitive. Competitive is simple, where you just have a molecule that fits the enzyme's active site and does not allow the substrate to bind. So that competitive inhibitor is just competing with the substrate for the active site. So you can imagine that the more competitive inhibitor you have, the more of it will block the enzyme's active site and make it hard for the substrate to bind there and then to have chemical reactions. So it slows down or it stops that chemical reaction. Non-competitive inhibitors simply bind to another site called the allosteric site, which is not the active site. But because enzymes are very sensitive to their to whatever's bound to them, when this non-competitive inhibitor binds to the allosteric site, it changes the shape of the entire protein. You can see it changes the active site so that the substrate cannot bind either. Now, we just looked at this, right? So this is allosteric control. We just looked at the allosteric site. That's why we have allosteric control, where we have an inhibitor coming in. When it binds, we can see that the active site is distorted and our substrate can't bind, so no chemical reaction. The opposite is true, though, with allosteric activation. So we have our same allosteric site. We have an allosteric activator at this point, and then the activator binds and it makes the active site even more functional, so it binds that substrate better. So we can either inhibit, slow down, stop the enzyme from functioning, or we can enhance the enzyme's function. All right, so the second part of this lecture is cellular respiration. So ATP is really important. It's the energy currency of the cell. To make ATP, there are two ways we're going to look at. One is called substrate level phosphorylation, which is using an enzyme. So in this case, the enzyme is called pyruvate kinase, and it takes a phosphate group away from this organic molecule, which is phosphophenol pyruvate, a big word. It's oftentimes abbreviated PPE, but this guy here just simply has an extra phosphate group, and it can be put on to our ADP molecule to make ATP. So what this enzyme does is it takes ADP, it takes PPE, holds them close together, and allows the transfer of the phosphate. So we have our product, which is ATP. The other way of making ATP is something called oxidative phosphorylation, which uses oxygen, which is why we have the oxidative there, and a process called the electron transport chain. Okay, so let's move on. We'll look at that. When we talk about the process of oxidative phosphorylation, that second way we make energy, we are going to be using oxidation reduction reactions, breaking down glucose. Okay, so glucose is C6H12O6. We're going to take glucose combine it with our oxygen, and then we're going to get, out of many, many, many redox reactions, the final product that we're going to have is going to be water. These are waste products, actually. Water is a waste product. CO2, carbon dioxide, is a waste product. And plus, what we're looking for, ATP. Okay, so to make ATP in this method, the electrons in this sugar molecule get transferred to oxygen. So in a redox reaction, we have redox means that we're just moving one electron from one molecule to another, and the molecule that loses an electron is oxidized. So that's our first oxidation. Oxidation means you're losing electrons. That process is called oxidation, right? And we have an oxidized donor. It lost its electron. The one that gets it, the electron acceptor, is going to be reduced. So if you gain the electron, it's called reduction, and you have a reduced molecule. So obviously, if you're going to lose an electron, something else has to pick it up. So if you oxidize something, you have to reduce something else. And that's why these always go hand in hand. So there's a fun little phrase, Leo the lion goes grr. So remember, Leo, lose the electron, you're oxidized. Grr, gain the electron, you're reduced. So in the overall equation for cellular respiration, the electrons here ultimately get passed to oxygen. So what do you say happens to glucose? Glucose loses the electron, so we're oxidizing glucose, and we are reducing oxygen, but in baby steps. So there are 3 main steps in cellular respiration. Let's go through them. Cellular respiration is very, very detailed in your textbook, and I just want you to know that this is all the information you need for the exam and for this class are the big ideas. And what is listed here is what you want to know. Glucose, which is a 6-carbon molecule, is converted to 2 molecules of pyruvate. Pyruvate are 3 carbon molecules. This is the process. You can see there's lots of steps to glycolysis. You don't need to memorize any of them, but we do need to know that the whole process here is anaerobic. So we can do this without oxygen. It does make some ATP. It makes a little bit of ATP with that enzyme, with substrate level phosphorylation. We also have a molecule we want to know. We're going to get to know this molecule very well. NADH is created. So here's our ATP. I think our NADH is over here. This is referred to as an electron carrier. Okay, it's an electron carrier molecule. There's 2 of these. One is called NADH and the other one's called FADH2. So remember that this is a redox reaction. So glucose has to move its electrons where? To oxygen, right? So coming back to this guy is going to get oxidized and then oxygen is going to get reduced. So in order to do that in little baby steps, there needs to be a molecule that takes the electrons away here and then ultimately gives them to oxygen. So that is going to be your NADH and FADH2. So let me just show you that when I like to call these guys Uber and Lyft because they're basically just driving electrons from one place to another. The empty molecule of Uber, right? The empty form is NAD+. When NAD+ picks up electrons, okay, picks up 2 electrons, it becomes NADH. Okay, so NADH, NADH. So let's think, NADH just gained some electrons. So it gained electrons, it's the reduced form. And it travels along with a little proton here with a little hydrogen ion, so this is the full form of it. This is our empty Uber, right? So this is able to receive those electrons. So when it receives, it gains those electrons, right? So this is going to be reduced. Now, ultimately, the electron carrier is going to move it to oxygen. We'll look at that in a little bit. Where this happens in both bacteria and in eukaryotes is in the cytoplasm. So when the glucose comes into the cell, it undergoes this process immediately. The second step is called the Krebs cycle, and it is a cyclical reaction, as the name suggests. What it does is it really removes all the carbon from our glucose. So remember, our glucose was a 6-carbon molecule, and all of our carbons are going to get basically released as a waste product, carbon dioxide. So we can see a couple carbon dioxides made here. Then we are also generating more NADHs and FADH2, right? So NADH is our Uber, FADH2 is our lift, and they're created in this cycle too. So we're taking electrons away from what's left of our original glucose, putting them into our cars, right? Putting them into the electron carriers, and we're going to move them to our third step. What we see happening is some ATP is made, again, with substrate level phosphorylation, just very little. And this is where you have a little difference, because in bacteria, this also happens in the cytoplasm. But in eukaryotes, because eukaryotic cells have mitochondria, and mitochondria is the organelle where we have ATP made, it happens in the matrix of the mitochondria. So if we want to remember what the matrix is, so the mitochondria has that outer membrane, it has an inner membrane, and the matrix is just the very inside of the mitochondria. Okay. One other thing just to note, right, this statement down here is that all of these steps in this Krebs cycle, some of these intermediates are used to make other things, right? So nucleotides can be made, amino acids, fatty acids. So a lot of products can be made from the intermediates here. In our third step, called the electron transport chain, this step requires a membrane. So we're going to have to find a membrane for all these proteins, right, embedded in it to be able to work. So in bacteria, where is the only membrane they have? Because remember, they do not have any membrane-bound organelles. Their membrane is the plasma membrane, so it's just their outer membrane. In eukaryotes, this happens in the inner membrane of the mitochondria, okay? The reason why membranes are important is because we want to create a barrier where we can have a concentration difference of our hydrogen ions. So on one side of the membrane, the bracket is for concentration of hydrogen ions. On one side of the membrane, we want a really high concentration of hydrogen ions. On the other side of the membrane, we want a really low concentration of hydrogen ions, because this gradient is going to power this guy, which is ATP synthase. The name is right here, and it's the enzyme that's going to make most of the ATP in this process. So this membrane is important. Hydrogen ions, just a refresher, hydrogen ions are polar, they have a charge, so they're not able to just slip through and go back down their concentration gradient, which is what they want to do, right? Diffusion states that molecules or just things at high concentration want to move towards low concentration until they reach equilibrium. But because of the charge, it can't pass through this fatty membrane. This is hydrophobic, this guy's hydrophilic, so this does not happen. So once the hydrogen ion gets pumped across the membrane, it stays here, it's stuck. So let's see what our Uber and Lift are up to. So the electrons, as we know, we want to pass the electrons to oxygen, because oxygen is going to be the one that gains the electrons or reduced, right? So NADH and FADH2 will drop off their electrons in the electron transport chain. When they do that, they become the empty form again, right? it becomes NAD plus, and then it also becomes FAD. So these guys can just drive back to the first two steps, to glycolysis and the Krebs cycle, and they can pick up more electrons. Now, in the chain, the reason why it's called electron transport chain is that the chain will basically bounce those electrons from one protein to the next. So the red arrow line you see is the route of electrons. So electrons will travel here, then to 2, 3, 4, 5, right? Every time these transmembrane pumps gets a hydrogen ion, gets those electrons, the electron will power this pump to pump that proton or hydrogen ion across the membrane. So you can see that happens 3 times. The electrons, as they travel, when it binds to this one, it powers the pump. So electrons, you can think of it as like a little bolt of electricity or energy. Then the electrons hit the third one, and then this guy does the same thing. It uses that little spark of energy to pump a hydrogen ion. So we have pumping mechanisms to create this gradient, right? We want a high concentration gradient on one side. Now, what is our final electron acceptor? Oxygen, right? So we see that right here. Electrons travel through the chain, but they're kind of like, okay, what happens now? There's nothing else to go to. It's oxygen. So oxygen is ready to pick up those last electrons that are at the end of the chain. And then as it does so, it will also pick up a couple hydrogen ions, just spontaneously form water. So water is a waste product, right, in our overall reaction. Okay, so let's go back and just look at our waste. So we have glucose, oxygen, the waste products of water. So we just saw water being made in the electron transport chain, right? That was when oxygen finally got those electrons and it just combined with some protons, and now we have water. This CO2, remember CO2? That was made mainly in the Krebs cycle, and that's basically the carbons, the 6 carbons here. We don't need them. It's really the electrons we're looking for. So the waste of the carbon is CO2. And now let's talk about how we make that ATP. Okay, what we've established is that the electrons are used to pass through this chain of proteins, and in that chain we have pumps that use those electron spark to pump hydrogen ions across the membrane. We've also said that these hydrogen ions are stuck. They can't diffuse towards the lower concentration. So why do we have to have this high concentration, low concentration? Because this enzyme here, which is called ATP synthase, very important enzyme, it really needs the movement of hydrogen ions flowing through it. You can think of this kind of like water behind a dam. If your hydrogen ions was water held behind a dam and you poke a hole in the dam, then the water is going to rush through that hole really quick, right? Same concept. We have a lot of these hydrogen ions. The only way they can get across, diffuse down into their lower concentration is through this enzyme. And this enzyme uses this rush of protons or hydrogen ions down itself and uses that energy to add that phosphate group. Remember, all we're doing is we're adding a phosphate group onto ADP. So when we do that, we make ATP. Okay. Turns out that this method of making ATP is very efficient, and we can get about 28 ATPs made per glucose molecule using this. Okay, so because we're using oxygen, this process is called oxidative, and because we're phosphorylating that ADP, oxidative phosphorylation. This is another look. This is from a different textbook, but it's the same thing. I just want to show you that this textbook shows you that bacteria have to have the electron transport chain on their membrane, and they basically pump those hydrogen ions into the space between the membrane and the cell wall. Whereas eukaryotes will have mitochondria, and their electron transport chain is going to be on the inner membrane of the mitochondria right here. You should know this difference, okay? That bacteria have it on their cellular membrane, pump it in that space between the cell wall and their membrane, and then eukaryotes have it on the inner membrane of the mitochondria. Okay, so the overview is in glycolysis, which is happening in the cytoplasm. We are making a couple of electron carriers. We're making a little bit of ATP. In the Krebs cycle, we didn't actually cover this, so I'm not requiring that you know that. In the Krebs cycle, we have made a lot of a lot of electron carrier molecules. We have that 2 ATP. And then we have our electron transport chain. Oh, glycolysis, Krebs. Oh yeah, so the electron transport chain it does not generate this, right? So the electron transport chain will actually generate at most 38 ATP altogether, right? So what we have is like 34 is sort of like the amount of molecules of ATP made by oxidative phosphorylation, which is the electron transport chain. And then theoretically, the most ATP you can get from one sugar molecule, we're just talking about one sugar molecule, is 38. And that's a really big number, okay? So this is the theoretical maximum, and they say theoretical maximum because living things are not perfect machines. And so we're not always going to get the same number of ATP per glucose molecule all the time. So, but this is like the maximum you can get. So let's answer some questions, right? The electron transport chain. What's the role of NAD plus and FADH? And how are these different from NADH and FADH2, right? So this and this, there are electron carrier molecules. These are the empty forms of them, and these are the full forms, so they're carrying the electrons here. So their role is to basically become reduced, right? Because they're going to gain electrons from our organic molecules, all right, beginning with glucose. So slowly, we're transferring those electrons to NAD plus and FADH. So they're reduced, right? And then once they come to the electron transport chain, what happens to the electrons from NADH and FADH2? Well, they're going to give them to the electron transport chain, right? The electrons are dropped off, they're donated to the chain. And so when they leave the molecules here, right, they lost the electrons, they're oxidized. So they become oxidized and they can be recycled to move more electrons. What waste product is made in the electron transport chain? Remember water, right? Water was made in the electron transport chain. What do the proteins of the electron transport chain do, and for what purpose? So by the proteins, I'm talking about all of this stuff, right? All of these proteins right here. I'm not talking about specifically this enzyme. This is the chain. This is the electron transport chain. This is just an enzyme that happens to benefit from the chain. So what do the proteins do in the electron transport chain? Well, they do transfer electrons, right? They transfer those guys from one to another. Ultimately, I'm just going to transfer, ultimately to oxygen. But in the process, they are going to create that really high hydrogen ion diffusion gradient, right? It creates a really high concentration gradient. So those proteins really shoot those hydrogen ions, remember, across the membrane, and they make that gradient. For what purpose? The purpose of these guys, why they're pushing those hydrogen ions across the membrane, is to make a concentration gradient. To make that concentration gradient, because why? Why do we need that concentration gradient? Because that's what ATP synthase requires. It needs that gradient so that hydrogen ions diffuse down it, and it makes ATP. So this answer to this, what does ATP synthase do? Simply, it makes ATP, right? What it does is it attaches a phosphate to our ADP, and then we can make our ATP. How does it get the energy to do that? Diffusion gradient, right? So the diffusion of our hydrogen ions. All right, some questions before we leave this section. Which of the following statements best describes glycolysis? Okay, if you look at it, it should be answer A. Which cellular process results in the most significant production of ATP? So we saw that glycolysis made 2. The Krebs cycle made 2. We haven't even talked about fermentation yet, but I will say fermentation makes 2. And oxidative phosphorylation makes 34. So obviously, this is the best way to make ATP. It's the most efficient way Where does oxidative phosphorylation occur in prokaryotes? Okay, so in bacteria, where's their only membrane? Cytoplasmic membrane. Which of the following statements regarding oxidative phosphorylation is true? Okay, if you have read all the choices, it's going to be answer C. Energy released when hydrogen ions flow down their concentration gradient, right, from high to low, and that's harnessed by ATP synthase. All right, let's stop there.